So just to remind you where we finished up last time, just to repeat the key point. When you have a number of replicators already in the DKS state, the dynamic kinetic stability state, the kinetic equations state explicitly that such replicators, if they feed off the same building blocks, cannot coexist. The better replicator will drive the poorer replicator into extinction, and that was already pointed out by Eigen and Schuster quite some years ago. And the good news is that in the test tube we see this behavior manifesting itself in this classic experiment by Sol-Spiegelman, though one has to say that one can see this direction of evolution, even though one has to state that the RNAs in the Spiegelman experiment were not in a dynamic kinetic stable state. We're just seeing the process of kinetic selection moving towards better replicators, but it does not fulfill the full conditions for getting a true dynamic evolutionary process. You'll notice here as well, it was done through 74 replication cycles. In other words, transferring constantly from one test tube to another just to see the process of evolution taking place. But the idea, the intellectual idea, was captured there. So what is the remarkable thing about this Spiegelman experiment? If you look at this sequence of events, replication, variation, selection, evolution, that sounds suspiciously like biology to me, the essence of biology. And here we're seeing that occurring at the molecular level. And this should give us some confidence that chemistry and biology are, after all, related, and that we can see that it's very likely, and we'll say more about this now. I'll try to give some, I've been asking in the first period more question than providing answers. Now in this second part I want to try and offer some answers to the questions that were raised. So one can see here that replication, variation, selection, evolution takes place at the chemical level and not just at the biological level. Evolution is also a chemical phenomenon for the right sort of replicating materials. So this is now specifying through that we have a driving force for evolution, and it is the drive towards greater stability, but not greater thermodynamic stability. This other stability, the stability of persistence, the drive towards greater dynamic kinetic stability, and it's one single process that starts with a simple replicating entity and goes all the way to complex life, the drive towards greater DKS, operating both in the chemical phase and the biological phase, one process. Okay, I said that already. The sequence applies to both chemical and biological phases. And that, I think, is already some evidence, not proof by any means, but evidence that life could emerge from the early Earth, because this phenomenon, which is the essence of biology, already can be seen to have its roots in chemistry. Now, if in fact the entire process is a single process, chemical evolution, biological evolution, it's one process, that has profound implications. It would offer new insights both into abiogenesis. Abiogenesis is just the process by which life emerged from non-life, and biological evolution. We'd learn about both from this fact that it is one. Why is that? You see, if it's two separate processes, then you have to learn the principles governing one and the principles governing the other. And that's what we've been doing till now. But if it's one process, what one can learn about abiogenesis may be relevant to biological evolution, and what one sees in biological evolution may be relevant to abiogenesis, because it's one set of principles for the entire process. So this could be a very powerful tool for understanding both parts. So we can learn about evolution from abiogenesis and vice versa. Now, I don't know how many of you are aware of this, but there are very deep arguments taking place within biology, between biologists, about neo-Darwinism, the gene-focused view of evolution, and there's an attempt by a growing group of opponents to say that something needs to be changed there with regard this approach, a more holistic approach to evolution. And I think something that is coming out from what I'm saying is that that one has now a tool to consider this, and actually it shows directions in which this can be pursued through understanding biology from the principles at the chemical level. Because you see, when you think of biology as just the genome, and it all starts from there, that neo-Darwinian approach tells us nothing about abiogenesis. In fact, that's one of its problems. If you have a theory of biology that tells you nothing about how biology came about, there's something lacking in that theory. So we need a way of looking, a theory of biology that not only tells you about biology, about evolution in biology, but relates it to the process by which biology came about from chemistry. It also means that when we talk about evolution and the general theory of evolution, we should express that theory in chemical and not biological terms. And let me explain why. In science, one always tries to have more basic explanations. If we're learning that chemistry and biology, abiogenesis and biological evolution are one process, then the terms that we need to describe that process have to be the most general terms, and then they cannot be biological terms. They have to be chemical terms. We can explain biology in chemical terms. We can't explain chemistry in biological terms. That's the wrong way to go. We explain chemistry in physical terms. You can't explain physics in chemical terms. The science has this hierarchical nature, and you can also always explain higher level phenomena with lower level concepts, not the other way around. So now maybe we should be able to try and describe or state evolution as a more general chemical phenomenon as opposed to a biological one. So the bottom line is abiogenesis is an amazing question, but it's not just an exotic corner of biology. I believe it would form the basis for much of biological understanding. If we understand abiogenesis, we understand how life emerged from non-life, we'll have a deeper understanding of what biology is all about. Because here we have almost a paradoxical situation that biology is the study of things that the biologists can't decide what they are studying. So a science that doesn't really know, it sort of has a feeling of what it's studying, but it can't specify what it's studying, because what life is still a question in itself confusing. But this unification may give us this broader, deeper insight into what is life. Okay, so I said it's a single process. If you say something like that, you better have evidence for it. And I just now want to point out the evidence, and I already gave one piece of information, but clearly if the evidence for the single process is that both the biological and the chemical phases exhibit the same or similar underlying patterns. And I already showed you one. This is just a repetition, that replication, mutation, selection, evolution occurs both in the chemical phase, the abiogenesis, we see that in molecules, and we see that in biological evolution. But now I want to point out a fascinating aspect that also unifies chemistry and biology, and that is the issue of complexification. You see, in this process from the simple replicating entity to complex life, something that happened the entire way along was increasing complexity. The systems started off simple and they became complex. Why is that? We have to understand that. First of all, is the fact correct that complexity increased? And the answer is, I think, pretty clearly yes. It's certainly true at the chemical phase, because a simple replicating system whose identity we don't know became simple life, and that I showed you is enormous increase in complexity, 9 orders of magnitude. So there it's quite clear. But in the biological phase, there's a little more argument here, and I don't really have time to discuss it. If someone wants to bring it up in a question, we can do that. But early life was prokaryotic, and then we got eukaryotic life, and this then continued on, evolved into multi-cell life, and of course the extensive ecological networks that individual life forms establish, which are a form of complexity. So this is all complexification. So there is clearly, in my view, even though some biologists, as I say, there's some disagreements here, but there is a clear tendency or evidence for a growing complexification within the biological phase of evolution as well as within the chemical phase. So the fact that this complexification operates both at the chemical phase and the biological phase is yet more evidence for the fact that we're talking about one process which manifests itself in the same way, increasing complexity. A very nice experiment from systems chemistry which shows the power of complexification at the molecular level is the following one, from the work of Joyce in 2009 in this wonderful science paper. He was looking for the best RNA replicator out of a large library of potentially 10 to the 14th RNAs, and he found it. It was just an RNA, let's call it T, that was able to make copies of itself from just 2 fragments, not individual nucleotides, but just 2 fragments. If you look carefully here, it's not that clear. Here is the T. This is T, the RNA with its tertiary structure. And here are the 2 fragments, A and B, and T, acting as the template, attracts A and B to it and makes another copy of itself. So T catalyzes its own formation. It's a replicating molecule. But for reasons that I don't want to get into, the reaction was not very good. It was slow, and there was limited exponential growth. It was there, but it was limited. It only did a couple of cycles, and then it stopped for kinetic reasons that we don't have to discuss. But this was the best one out of this huge library, okay, of 10 to the 14th. But then he did something else. He sought what happens if he takes 2 RNAs that cooperate and maybe can replicate in a different way and through a cross-catalytic process. So the 2 RNAs that he took now, let's call them E' and E. Now E' is not good at making more of itself, and E also can't make more of itself, or not very well, but E' can take the components of E and make a copy of E. So E' catalyzes the formation of E, but E is able to catalyze the formation of E' by taking the components of E'. So instead of having a single template process. You have a little network where E prime makes E and E makes E prime, and that creates a replicative cycle. Through cross-catalysis, we have autocatalysis, because if E prime makes E and E makes E prime, the whole system is making copies of itself. And here is the important thing. Once he took two molecules out of this big library, suddenly the replication was much faster, and it led to self-sustained exponential growth, provided that building blocks were supplied continuously. So what's the conclusion from this? It seems to be that complexification enhances the replicating ability at the molecular level through network formation. A more complex replicator is able to do the job better than a simple one, and I sort of have a simple analogy to this. If any of you eaten Chinese food, you will find it's a lot easier to eat Chinese food with 2 chopsticks than with 1. That simple idea is actually hidden within this result. So the complexification principle might be specified as I'll scratch your back if you scratch mine. Horses have evolved. They're one of the, not that many animals can't scratch their own backs, but horses' legs have evolved for running and not for back scratching. So if horses want to scratch their backs, they have to do it to one another, as these 2 horses seem to be trying that here. So the 2 horses here are doing, in a sense, what these 2 molecules are doing in the molecular network that Joyce showed. We can consider the chemical term complexification equivalent to the biological term cooperation, but the net result is that whatever you call it, enhances the replicating ability both at the chemical and the biological levels. And we saw the chemical one through the Joyce experiment. And I put it here again, not to forget to say it, network formation, how important that is, network formation. And the more you look in biology, you see networks, because networks improve function, and I'll be talking about function pretty soon. So then the difference, we said it's one process, from simple replicating to complex life, it's one process. The difference between the chemical phase and the biological phase is just the level of complexity. The chemical phase is the low complexity phase. The biological phase is the high complexity phase. Again, one process towards greater stability, low complexity, low stability, high complexity, high stability. And then when we talk about this transformation from simple to complex, we can suddenly from that conclude that the evolutionary process, we tend to think of it traditionally in terms of what's evolution about? Natural selection. That's the essence of Darwin, natural selection. But now we're seeing, and others have pointed this out, that actually there are 2 pillars, important pillars to the evolutionary process. One is the selection, but the other is the complexification. So if we talk about it in chemical terms, kinetic selection and complexification are very important for evolution. The biologists might call it competition, the natural selection, and cooperation. The 2 together, and that's how we got from this little thing to this big complex thing through, let's call it molecular cooperation. The molecules cooperate to create this very effective replicator. You see, this is a terrible replicator. If you want this to replicate, the molecule, you need a lab students, grants, otherwise, and then if the molecules are in a bad mood, they won't replicate. This thing here, outside, in the soil, it doesn't matter if it's raining or sunny day, replicate no problem because of its complexity. The complexity has made it an extremely persistent and capable replicating entity through complexity. If you want to convince yourself that life is complex, this is a map of metabolic pathways, and the next 5 hours I'll go through each of these little reactions in detail. You don't want me to do that, but that's just a picture of metabolic pathways to show you how complex, how much complexity there is in the chemistry of life. So that's in the reactions. It's also clear in the structure of some of the organelles. The ribosome. The ribosome is an extraordinarily complex structure. Basically, you have two subunits made up of several RNAs with protein in as well. But the molecular weight there is roughly 2.5 million Daltons, a huge structure, and of course the Nobel Prize for elucidating the structure of the ribosome was just given out some years ago, 2009, to the people who did this very impressive work on elucidating this complex, very complex structure. The central dogma of biology, we're all familiar with this, DNA to RNA to protein. That was the great idea of Crick many years ago. But actually we see that things are more complicated than that, and in a sense one can present it in this way, that of course the protein is not the end of the road. You need protein to make protein, you need protein to make RNA, you need protein to make DNA. This is a highly complex network of DNA, RNA and protein. That's what biology, functional biology is about. And so it's not just this one-way thing that is maybe oversimplified in the central dogma. But if life is complex, and we said evolution went from simple to complex, there must have been simpler phases or stages along the way, and the RNA world is exactly the proposal that says that initially the life would have been more based on RNA, where RNA acts not just as the informational molecule but also as the catalyst. RNA can be a ribozyme, and of course that was the work of Altman and Czech, and they received the Nobel Prize for that. And it's, as I said, that there was an RNA world that preceded the nucleic acid protein world. Now the beauty is, you know, evidence for that, the ribosome structure that I just showed you is a nice piece of evidence that lends support to the RNA world idea. And I want to show you the the ribosome picture again, but in a different way. Here it's shown where the RNA is shown as silver, and the protein is shown, the protein components, in gold. And where the ribosome does its actual stitching together of the amino acids to make protein is in the center. Now if you look at that structure, you can see the central part of that structure is all RNA. And the protein segments are more on the outside. There's a message in that fundamentally the ribosome is an RNA machine, and that it did its translation as an RNA machine which did not need protein in order to function, Proteins subsequently got incorporated into that structure, but for more to improve its function, for fine-tuning, so it wasn't essential to that RNA, to that evolutionary process. So the point is that just the structure of the ribosome itself seems to tell us that initially we did have an RNA world, and slowly the protein world became part of that world through a process of complexification, which led to improved function. But now we have to address the question of complexity. Why complexity? We've said the evolutionary process is towards greater complexity, and in some way, for some reason, this greater complexity is leading to greater stability, but stability in the sense of persistent, not stability, not thermodynamic stability, greater DKS. Why complexity? Where did function come from? I already mentioned this, I think, in one of the questions, we discussed it. In the non-living world, there is no such thing as function. Atoms and molecules don't have functions. Mountains don't have function. You just have the laws of physics and chemistry, and everything does what it should do based on those laws. But in the biological world, function is everywhere. It's only in the biological world, and just if you want proof of function, here is a heart. You know what its function? It's to pump blood, and this is an ear for those who don't recognize it, and its function is to listen. And here is the ribosome with its function to make protein. The three functions, there are just zillions, endless functions in biology. Biology is all about function. Where did function come from if biology came from chemistry and there's no function in chemistry? A puzzle. And I think the answer is the origin of function is in this very simple transformation. Poorer replicators tend to become better replicators. That's a law of nature. We saw that. It's in the math of the replicating equation. From less stable, DKS, less stable, to DKS, more stable. Now, we take that for granted now. Replicators become better, but this is a very unusual change because there's no law in science that applies to other things, other characteristics. Poor acids, there's no law that says poor acids will become good acids. Poor oxidizing agents tend to want to become better oxidizing agents, etc. In the regular world, it's all the second law. Nothing about worse becoming better. It's only in the replicative world that we have a process in which, for mathematical reasons, kinetic reasons, poorer tends to become better. And that creates the function of replication. Replication becomes a natural function, the function of replication, and then anything that will facilitate that function is seen as functional. It facilitates the function of replication. So this is a beaver dam, not too clear. It has a function. It enables the beaver's nest, its home, to remain covered by water so that the beavers can live without having to worry about predators. This is a termite mound, an inanimate object again. It has a function built by it, so it's not just these organic biological entities that have function. Inorganic or non-biological entities can also have function. So this has, its function is to provide a house to the termites so they can live better, replicate more effectively. I guess presidents need a nice house too so that they can hopefully function better. And of course, any inanimate object that is created by living systems improves function, and it's only all function has a biological context. But okay, so now I want to try and understand, back to the question, why complexity? And I argue that there is a relationship between function and complexity, and I think it's most simply described by this transformation here. This is the Wright brothers' plane in 1903, and it wasn't a very complex plane, but it wasn't a very functional one either. I'm not sure too many of us would want to fly transatlantic in one of these. And then over 60 years it became that, okay? Boeing 747, very functional, but very complex. The complexity facilitates function. And remember the business with the chopsticks? When you have more components, you can do the job better. Complexity is a very, very, very difficult concept to define, but in simplest terms, the number and diversity of components. But as soon as the moment natural function emerges through the replication process, nature begins to act as an engineer, because it goes from poorer functioning, poorer replicators to better replicators, so it necessarily acts as an engineer, and that is the beginning of complexity and what one might call apparent design. I don't want to call it design because we know design is like deliberate. And but to show you that deliberate design and apparent design are not too different, here are just two buildings, a temple, Angkor Wat, in the Angkor Wat complex in Cambodia, and here's the Gaudi Cathedral in Barcelona, built through human design. Here is a termite mound built by termites. So you can see, we think we're very different to other species, but maybe we're not that different after all. So I see here, if you like, a triad of complexity, stability, and functionality, all directly related. Functionality is the essence. We want a better replicator, a more functional replicator, and in order to make the function better, it has to be more complex, in that, according to that same principle, that improved function requires greater complexity. But of course, that improved functionality comes about, because it's leading to greater stability. That's what the function is. Greater stability, but stability in the replicative world, dynamic kinetic stability. So we have here immediately a connection between complexity and stability. In other words, the more complex is needed for greater stability. I have to be careful here not to imply that complexity in itself leads to stability. That's not true. You can't just make something more complex and for it to function better. You don't just put on a plane, you don't put just another knob on the wing to make it more complex. That won't make it fly better. It's got to be specific complexity, functional complexity, for it to operate. So we have here basically this connection between complexity, dynamic replicative stability, DKS, persistence, if you like, in the replicative world, and functionality, greater functional complexity for greater stability. So if there's a natural tendency towards greater stability, then it's brought about through greater complexity. And that maybe explains why this statement here, if you want extraordinary function, And there's no question that the function in the replicative world is extraordinary when you look at the bacterial cell with the incredible, I mean, the more we look at it, the more we're amazed by the complexity, the intricacy of what goes on in that tiny entity. But that extraordinary function comes about because of the extraordinary complexity. Simple function requires simplicity, but if you want very good function, you have to have considerable complexity. This leads me to an interesting question that only a physicist, a Nobel Prize physicist like Erwin Schrödinger would think of asking. Why are atoms so small? I mean, only a physicist would ask a question like that. But then he went on and he said, clearly the question is an evasion, for it is not really aimed at the size of the atoms, it is concerned with the size of organisms, more particularly with the size of our own corporeal cells. The question he was really asking is, why are living things so big in terms of atoms and molecules? And here I want to offer an answer to that question, because extraordinary complexity is essential for the extraordinary function and stability associated with living things. Okay? I've been talking about extraordinary function. Biological entities just are remarkable in how they can do things, leading to that extraordinary stability, and that requires extraordinary complexity, which in simple terms is size. You need lots of stuff in order to bring about that function and stability. So this leads to a general theory of evolution, which basically says that all stable, but stable in the sense of persistence, not in the sense of thermodynamics, all stable, persistent replicating systems will tend to evolve primarily by complexification towards systems of greater stability, but this other kind of stability, dynamic kinetic stability. And of course, as I said, the theory, the extended theory would embrace both biological and chemical systems. And then we see that Darwinian concepts that we're also familiar with are just particular applications of broader physical and chemical concepts. So we're all familiar with natural selection, fitness, survival of the fittest, cooperation. And now we can see that there are physical, chemical concepts that are at the root of these biological concepts. So basically natural selection is just the expression of kinetic selection in biological terms. Fitness is dynamic kinetic stability. Survival of the fittest is the drive towards greater stability, but this other stability. Cooperation is complexification. And that translation is important because, as I say, it then becomes more general. If we just use the biological concepts, then it seems inappropriate to talk about survival of the fittest with regards molecules. And we don't talk about the fitness of molecules, and molecules don't cooperate because, you know, there's no agenda in the world of molecules in chemistry. But as I say, biological concepts firmly rooted in physics and chemistry. And now you can see what I'm driving at all along. Biology, a complex manifestation of replicative chemistry. That's essentially the key idea. Now, living things have remarkable characteristics, and it's been very hard, so different to inanimate objects, and it's been very difficult to explain those characteristics in chemical terms because, well, it didn't work very well because of the special nature of biological systems. But I think through the DKS concept, a lot of the characteristics can be explained and understood in a better way. I've talked about functional complexity. I discussed that. How can there be function, and why is the function, why is there complexity? We discussed that. And now I want to discuss this issue of dynamic character. I saw, I think I saw Muriel in Porto, when was it, in May? In May last year. And I have to tell you with some sadness that the person sitting here is not Muriel. It's an imposter. I mean, Muriel of last year there's certain atoms and molecules that when they come together, that's Muriel, right? That's what defines Muriel as Muriel. But all of those atoms and molecules have been turned over. It's already different stuff that makes our Muriel today. So the question is this Muriel if it's different stuff from last year's Muriel? I mean, isn't something defined by what it's, the material that it was made of? And this is a very puzzling thing, that we're changing, we're turning over all the time. The matter that makes us us is changing. I'm not sure entirely of the time frame, but certainly within a period of some months, much of us has changed. And it even applies to things like our bones. I mean, you think of bones, when a bone breaks, the bone heals because there is a process in which the bone is, I don't know what's the word, not dissolving, that's not the right word, but there's a constant regeneration of the material that even makes up our bones. So, and I'm not talking now about the molecules in the cells, which are constantly turning over, and I'll talk about that in a moment. So how can this, you know, life is a very strange phenomenon if everything's turning over all the time. So what's the explanation for that? And there it is. Remember the fountain? That's the nature of fountains. The fountain is persistent. It's still the same fountain that was in Geneva last year. But, of course, the water in the fountain is different. It's turning over. And in the same way, we are a dynamic system in which the stuff in us is turning over. We have here stability in the sense of persistence through change, which almost sounds like a contradiction. But it's the change that facilitates, enables the persistence to be there. And interestingly, the steady states that we talk about exist at different levels of complexity, and I've said there's this tendency towards greater complexity. If we're talking about a population of molecules, molecular replicators, there's just one level of turnover. It's just the fountain picture that I showed you. But when we go to the cellular level, we have two levels of turnover. The molecules in the cell are constantly turning over, and that was actually shown explicitly for the proteins. Protein degradation and resynthesis is a tightly regulated process in the cell. Intracellular protein has a half-life of minutes to some hours. So, and this is important for biological function. So there's a constant turnover of the molecules in the cell, and Nobel Prize was given for that, as shown here in 2004. But of course, the cells themselves are turning over all the time. So it's a, if you like, a fountain within a fountain. When you have living cells, the cells are turning over, they die and new cells come about. And while the cell's alive, the molecules in the cell are constantly turning over. And at the organismic level, us, it's a triple fountain. The molecules in our cells are turning over. Our cells are turning over. Every day we lose millions, billions of skin cells, blood cells, I hope not too many brain cells, but some of those too. Cells are turning over constantly, and of course here's the bad news. We are turning over as well, but the time frame is a little longer, happily, for that. But that's the nature of the process, turnover. So at the organismic level, 3 levels of turnover. The far from equilibrium state. This is it's a thermodynamic puzzle why unstable things persist. But, and I've tried to discuss that in showing that having a different kind of stability, not the stability, thermodynamic stability, but the stability in the sense of persistence, leads to dynamic kinetic stability, this other stability kind, replicative stability. And of course this dynamic kinetic stability depends on thermodynamic instability, because replicating things, in order to maintain themselves, have to be reactive. They have to be making more of themselves, and if they're going to be stable thermodynamically, they won't make more than themselves, they won't be reactive. So you need thermodynamic instability, you need reactivity in order to make dynamic kinetic stability, almost a contradiction. So life systems have to be far from equilibrium in order to maintain the fountain, to keep it going. So life's far from equilibrium state is an energetic prerequisite for its existence. And finally, the diversity issue. Life is extraordinarily diverse. The billions of different species that appear to have been produced over time it's just so different to what we have in the regular material world. Why is there such diversity in the biological world? I read recently that in your navel there may be 1,400 different bacterial strains sitting in your navel as we speak. Okay, so biological diversity is just one of the characteristics of this other world, the biological world. I think if we think about it, the topology of change in the two worlds, the regular world and the replicative world, are different. The thermodynamic and the dynamic kinetic world. The thermodynamic world is a convergent world in that all systems, chemical, physical systems, are directed towards their thermodynamic sink. So if you have a number of isomeric forms, they all want to end up in one place, the most stable form. So there's something convergent about the thermodynamic processes. But in the replicative world, in the world of dynamic kinetic stability, the space is a divergent one, in the sense that there is no clear direction that leads to greater dynamic kinetic stability. It depends on the circumstance. So in one situation you might be better off going this way and you'll create something more stable, but in a slightly different situation you're better off going that way. So the replicator space is inherently divergent because dynamic kinetic stability doesn't just depend on the system itself, but on its environment, because it's the environment that determines whether that system is stable or not. A bacterial population in a pool of water will be stable until one pours in disinfectant. You put in disinfectant, you've changed the environment, you've changed the dynamic kinetic stability. So it's a divergent path, and that different topology explains the diversity. In a sense, if you look at that divergent space, I won't talk about that. It also there's some issues there with Darwin's principle of divergent that fits in there, but I'll just skip that. And just remind you, the phylogenetic tree of life, its divergence is, as I say, a function of the nature of dynamic kinetic, the space, the divergent space of dynamic kinetic stability. It actually leads to an interesting consequence, and that is, in the regular world, because the space, the thermodynamic world is convergent, history is inaccessible, but the future is predictable. We know where things are going because they're all headed towards the thermodynamic sink. But if you want to go back in time, you can't tell where you came from because going back in time, it's divergent. Okay? But in the replicator world, the topology is the reverse. So we can actually look back at our history, and that's why we can build up the tree of life. So there we can access the history by looking at that tree of life, but the divergent nature of the space means the future is unpredictable. So if you want to know where we're headed, the answer is we don't know, but I think it comes about through this the different topologies in the two worlds. But now, finally, in the last moments, I want to talk about some work that I did together with Robert Pascal, where we've tried to take this a little bit further and try and unify to show the connection between, in a clearer way, between dynamic kinetic stability and thermodynamic stability. It's uncomfortable to have two stabilities. Can we make one, can we unify this so that it all holds together as approach to, well, you have this stability for the non-living world, this stability for the living world. There's something a little bit arbitrary about having two stabilities, and we're quite excited about this new development, which, as I say, was just published, actually, the main paper some weeks ago, or maybe it's a month or two now, in Chemical Communications. Again, what is stability? Stability, we're saying something is stable if it's persistent, unchanging over time. That's the fundamental meaning of stability. Persistent, unchanging over time. But it has two facets, as we've been discussing. There's the energy entropy facet, and there's the time facet, and these two aren't necessarily, they don't necessarily lead to the same thing. That's why we have thermodynamic stability, which focuses on energy entropy, and the dynamic kinetic stability that focuses on the time persistence. But can I connect these two together into one? Is there an underlying connection? And I think there is, which can be shown in this very simple Venn diagram. The Venn diagram shows that thermodynamically stable systems are a subset of the general set of persistent systems. So persistence is about time, thermodynamics is about energy. It's a subset. Why is that? Thermodynamically stable systems are persistent. Once you've reached the equilibrium, nothing happens anymore. It stays unchanged. So once you've reached thermodynamic stability, you've reached the persistent forms. So for that reason, thermodynamically stable systems are sitting in this big persistent systems area. However, persistent systems aren't necessarily thermodynamically stable. And we showed you, I've been discussing how dynamic kinetic stability is sitting in here, not in here. So it's obviously a persistent system, but separate from the thermodynamic stability, this thermodynamic subgroup. So what that means is that persistence is the more general criterion of stability, more general than energy slash entropy, persistence. Again, because of this Venn diagram, that all of the thermodynamically stable systems, energy, entropy, are sitting in this bigger group. So there seems to be a general rule governing change in the world which says things tend from less stable in the sense of persistent to more stable in the sense of persistent forms. And one can simplify that just to, in few words, nature seeks persistent forms as a general underlying principle of nature, which, if you think about it, is axiomatic. It's almost true by definition. And let me say why. You'll all agree with me that unchanging things don't change, and changing things do change. That's a tautology. Until they change into things that don't. So just look at that again. Things that change, change until they change into things that don't. So there's a tendency for things to become more persistent as a logical statement about change. So persistence is a very important, this kind of stability is a very fundamental kind of stability, as I say, which is logical in nature, and one, maybe this slide is not entirely in place here, but if we ask which stability is greater, dynamic or dynamic, kinetic or thermodynamic, you know, it's a little bit comparing apples to oranges, but you might think that thermodynamic stability is more substantial, more greater, intrinsically greater than dynamic kinetic stability, which is changing all the time. But just a number that might kind of make you think twice about that. How stable is Mount Everest? And the answer is very stable. It's existed for 60 million years. But remember those cyanobacteria that we talked about, morphologically largely unchanged for several billion years. Of course, it's not the same cyanobacteria. They're turning over all the time, but the population is there over extended periods of times. So this persistence can, even though it's in a non-thermodynamic context, can be very great. And where is this leading us to? It's that there are two evolutionary paths to persistence. There are two ways that nature can seek persistent forms. There's the way it does in the regular world, which is toward thermodynamic stability based on probability theory, and in the replicative world, through exponential growth and dynamic kinetic stability, another way of reaching persistence. Two different ways of reaching persistence. One manifests itself through increasing entropy, and that's the second law of thermodynamics. And the other is through increasing complexity, and one might say it's almost a second law analog. Okay? So in the regular world, you get to more persistent forms through increasing entropy. In the replicative world, you get to more persistent forms through increasing complexity. But in both, you have an evolutionary process of change leading to more persistent forms. So evolutionary change takes place in both worlds, the regular world and the replicative world, except that in the regular world, we don't call it evolution. We call change in the regular world, we call it change. That's the same word. And in the biological, in the replicative world, we have this fancy word for change called evolution. But there's an evolutionary process in both worlds towards increasingly persistent forms, once in one pathway a la Boltzmann, and the other pathway through the exponential growth pathway. And importantly, both processes are underpinned by math. So you have two mathematics that lead to persistence, and therefore two material forms. One might, hypothesize that if there would have been three mathematical ways of formulating persistence, there could have been three kinds of material forms. Why are there just two material forms, living and non-living? Why not three or four? I'm saying there appear to be two material forms, very different in how they behave and how they operate, because there are two mathematical ways of achieving persistence: Boltzmann's way and Malthus's way. So, in a sense, what we've been trying to do here is reduce biology to mathematics and logic. We're all familiar with natural selection as a biological principle, and initially we can actually reduce this and see, it's actually kinetic selection, which is a chemical principle, and then take it another step further to dynamic kinetic stability as a physical chemical principle, talking about a different stability kind. And now, taking it another step further to this general mathematical, logical principle, nature seeks persistent forms, and that biology or replication is just one way of achieving persistence. So in that sense, the conclusion is rather staggering, actually, that life, instead of being something inexplicable, is actually a logical phenomenon, because it's just a way of achieving persistence, but through the replicative pathway based on the math of replication. Okay? That almost suggests that life should be a very common phenomenon in the universe. That's the implication, but not necessarily, because we still don't know enough about the replicating process and the ease by which inanimate systems can achieve or generate a replicating state that can start off on this path. We know nothing about that really, and that's why, in some sense, we're at the beginning of this long journey as opposed to the end of it. But what I'm very excited about is, remember I spoke to you about, here we have the second law of thermodynamics, everything goes downhill, and Darwin, everything goes uphill. Somehow incompatible. Now, by looking at nature seeks persistent forms, one can see that you have a unifying principle for the second law and Darwinian natural selection. It sits above, if you like, both the second law and natural selection, and can bring Boltzmann and Darwin into harmony through this simple idea, which is logical. It's not, you don't have to do a check on it. It's a thought experiment. Nature wants to, things that persist will persist, and those that don't persist won't persist. So through this idea, both the second law pushes towards persistence in one way, and natural selection, if you like, the Darwinian idea, pushes towards persistence in a different way. So they're both in harmony in seeking to reach persistent forms. So I want to now, a little bit earlier, but I'm pretty well at the end there, to summarize what I've been saying. Well, to address, before we do that, the why question, I said, is there a why to the origin of life problem? Was there a driving force? Is there a physical basis for the process? And my answer is a definite yes. The drive towards persistent forms, persistence through replication. Okay? So now the conclusions that I want to make. The how and the where questions, historic questions, and in the absence of a historic record, we may never really know exactly the pathway and the location because the historic record has been eliminated. But the why question is an ahistoric question. It doesn't depend on looking for historic evidence. And the why question is the general one: why would inanimate matter be driven toward life? And I've tried to give some indication as to why that would come about, and that opens up hopefully a means of making proto-life, if we understand the principles by which that process came about. Because I think through answering that question we maybe have a better idea of what is life, and maybe in simplest terms, very simplest terms, a dynamic, self-sustained replicative network. And life on Earth is a dynamic, self-sustained replicative network based on the protein-nucleic acid duality, but that's not to say that that is the only chemical means of achieving a sustained, self-sustained replicative network. There may be other chemistries that we're unfamiliar with that could do that, and we have no idea because nothing along these lines has been explored. So that is the challenge today, I would say, to create a dynamic, self-sustained replicative network. But what it's saying here is that if, as some people have been trying to do, we want to take the components of life, put them together and hope that this will suddenly be alive, that's unlikely to work, in my view, because you have to have this far from equilibrium, dynamic, replicative state. You have to have a situation where you're creating your replicators and they're decaying, so you create this, if you like, fountain, and then you're in a process of being able to even watch this particular system evolve as it undergoes change of some variation. So that's the challenge, to try and make something that is starting to show some of the real life characteristics, and this is a quote from Richard Feynman. This was found on his blackboard at the time of his death, actually. What I cannot create, I do not understand. I've tried to show you that we're getting closer, maybe, through some of these ideas to an understanding of what is life, but the ultimate test will be to go into the lab and to do it, to make something hopefully based on some of these ideas that I've been describing to you. Muriel mentioned, I can't not, since this just came out literally, I only received this some days ago, a new edition here of some of these ideas, updated with this new material that I've just been discussing, because that new material doesn't appear, of course, in the original book in 2012. So some of this is now written here in this What is Life book with Oxford. And acknowledgements. Very important for me to acknowledge a wonderful collaboration I've had now for a number of years with Robert Pascal in University of Montpellier. Really a wonderful collaborator, and his great understanding of these issues. If you want to talk to him about physics, that's fine. Chemistry, that's fine. Biology, that's fine. He seems to understand all of these things, and that's great to work with him. Other people with whom there's been collaborative work leading to some papers, Nathaniel Wagner and Emanuel Tannenbaum at BGU. The late Emanuel Tannenbaum, I have to say, very sadly passed away recently. And John Sutherland from Cambridge, and of course Cost Actions 1304 and Murals 1308, Origins for the ability to communicate and exchange ideas, which is obviously a very important part of the process. And some pictures of where this work has been done. Thank you very much.